Yes, good morning, children. Let's go further in the structure of proteins. We have to understand the next classification, that is primary, secondary, tertiary, and quaternary structure. Okay. Now, you know very well that proteins are nothing but polypeptides. Now, polypeptides, a peptide, how it is formed? It is formed by the combination of two alpha amino acids through an amide linkage. So the amide linkage present in between two alpha amino acids is called as a peptide bond, right? Okay. Now when these peptide bonds, they become many between too many amino acids, when it is combining together to form a long chain, then you call it as a polypeptide. Now, what is this primary structure? Primary structure describes the sequence of amino acid arrangement in a protein. Which amino acid will come first, then what is next, then third, what is there, fourth, what is the amino acid. So that sequence of alpha amino acids to form a polypeptide chain is what the primary structure describes. Now, any change in the amino acid sequence results in an altered protein, which may or may not have any significance in the body. Now, this is what is happening in the case of normal hemoglobin, and you must have heard about sickle cell anemia. So sickle cell anemia patients, they will have their hemoglobin in what sequence? Just see. And one change in amino acid in the whole polypeptide chain, how it is totally changing everything. You have valine, histidine, leucine, threonine, proline, glutamic acid, again another glutamic acid, and then lysine. Now in sickle cell anemia, everything is same. Instead of glutamic acid, valine is coming. So this one change in amino acid in the sequence is totally making the hemoglobin, which is malfunctioning. Okay. So this is what is to be understood. Now first ever primary structure of a protein was given by the scientist by name Frederick Sanger for insulin. Insulin is a protein. It is also a hormone. Okay. And he won the Nobel Prize in the year 1958. So this is about primary structure. So what does it tell you? It tells you the simple sequence of amino acid in a polypeptide chain, which results in the formation of a protein. Now, what is secondary structure? Secondary structure arises due to the polypeptide chain, which undergoes some curling around. The conformation, which arises due to hydrogen bonding in polypeptide chains, results in secondary structure. Now, this is of two types. What are the two types? The two types are alpha helix and beta pleated structure. Now let us see what is alpha helix. The coiling of the polypeptide chain into a spiral structure in a right-handed form is called secondary structure, is called alpha helix. Okay. Now this alpha helix was postulated by the scientist by name Linus Pauling in the year 1951. Now how it is turning, see. Supposing you consider this as your thumb. So thumb का ये axis है, ठीक है? This whole line that is forming your thumb, that is following the axis, and the turn is taking place like this, a coil around the axis. So this is the axis, so it is coming around like this, right? Now each turn of the helix has 3.6 amino acid residues and a 13-membered ring through hydrogen bonding. So I have given another video for you to make you understand this even more clearly. Okay, so please look at that and you will have a thorough understanding. So this is how it is coiling around the axis, main axis. Okay, now you have all amino acids, that is your polypeptide chain is moving around like this. And what you find will be, supposing if there is a CO group and here there is NH group, and between these two hydrogen bonding will be there. Okay, try to understand, they are concentric circles, it is coming around like this. So like that there are so many intramolecular hydrogen bonding which holds up the molecule or the polypeptide chain in this alpha helical form. And distance between two successive turns, see, this is called as the pitch. This distance is 54 picometer. Okay, now why, you may ask, only right-handed helix, why it is so, why right-handed? Because in this form only there is maximum number of hydrogen bonds. That is why this alpha helical structure has been found to be the most stable one, right? And then the next one is beta pleated structure. Now in this beta pleated structure, what happens? The polypeptide chains, they are laid side by side. They are laid side by side. This is one polypeptide chain, this is another polypeptide chain. Okay. Now this beta pleated structure is again of two types, parallel and anti-parallel. Parallel means one polypeptide chain, it is starting with N terminal and it is ending with C terminal. That is, it is starting with amino group and then it is ending with a carboxylic acid group. Same way the next chain is also the same way, N terminal and C terminal. Whereas anti-parallel में क्या होगा, one chain will be N terminal chain ending with carboxyl group and the other one will be C terminal ending with N terminal. Then the next one again N terminal with C terminal. So that is anti-parallel. One chain alternating with the other in terms of the terminal positions. Okay. Now parallel में क्या होता है, ये N terminal है तो next chain भी N terminal है, third chain भी N terminal है. So all these chains, they are arranged side by side like this. Okay. Now in this what will happen, supposing if there is a, see, this is NH, then CHR. Now you must be knowing what is a polypeptide chain. NH, CHR, then CO, again NH, again CHR, again C double bond O, NH, like that it is what? Continuing. Now the CO group of this chain is attached to NH group of this chain through intermolecular hydrogen bond. So this is what you call it as parallel structure. In anti-parallel, it is just the opposite, that's all. Is that clear? So that is what is called as beta pleated structure. So इस तरीके से arrange करने से क्या हुआ? You will find that here there is a CO-NH bond के बीच में वो intermolecular hydrogen bonding आ गई और यहाँ पे next वाले में यहाँ पे आ गई, फिर third वाले में यहाँ पे आ गई, फिर fourth वाले में ऐसे आ गई. तो when you fold it, you will find it in the form of pleats. Like जैसे साड़ी में pleats बांधते हैं तो वैसे वो pleated structure देगा. Between the folds, these hydrogen bonds will be there. Exactly, that is why it is called as beta pleated structure.